Okay, so chapter 33 is titled Animal Diversity and the Evolution of Body Plans. So I'm going to go through 9 characteristics of animals and then talk about how the animal body plan evolved. Then we're going to visit phylogeny again, and then start off with parazoa, animals that lack specialized tissues, and then move into true animals, animals with true tissues, and then start talking about bilateral. So here we go. Animals do share some general characteristics, many of them, as you saw in the guided notes. Heterotrophy, how they obtain energy. We have herbivores, carnivores, omnivores, detritivores. Multicellularity, all animals here are going to be multicellular. No cell walls of any kind. So the cells here are flexible. They're held together by collagen or some type of extracellular matrix. Active movement. Okay. So in some stage of their life, there is some movement. Diversity of form and how they range in that size. You know, animals, they're grouped in between 30 to 40 different phylogenies, and most of them do occur in the sea, and you'll see that. And then, diversity of habitats. Sexual reproduction. Embryonic development, where we have the combination of gametes for fertilization. You have a zygote. It goes through multiple rounds of mitosis, cleavage, and then blastula, blastropore. And then the fate of that blastropore determines whether or not you are a protostome or deuterostome to a gastrula, which helps us form either 2 or 3 layers inside the organism. And then finally, tissues, which just means a group of cells that carry out a specialized job. And sponges here are the exception. Sponges do not have true tissues. I just want to visit sexual reproduction. So we talked about plants in the previous unit, how plants go through alternation of generations. They have a multicellular haploid state, a multicellular diploid state. You don't see that here. So there's no alternation of generations. Okay, moving on to the review. Which of these is not a feature common to all animals?
E.
Yeah, I'd say E.
E again, it's like right there.
Which of the following is a defining characteristic of an animal?
E.
Match the type of heterotroph with food source.
B.
It is B. Herbivores consume autotrophs.
Wow. What a guess.
All right, moving on to body plans. So I'm going to talk about body cavities and the 3 different types of body cavities. Differentiate between a pseudocoelom and a coelom, and then talk about protostomes and deuterostomes, just a little preview and advantages of segmentation. So there are at least 5 key innovations in animal evolution. First off is symmetry. So we will talk about radial symmetry and bilateral symmetry here. Tissues, allowing specialized structures to carry out specialized functions. Body cavities, whether or not they have one or kind of do. And then various patterns of embryonic development. So we'll talk about spiral cleavage versus, I'm blanking on the other one, I want to say it's radial cleavage. And then this idea of segmentation or repeated body units and the advantages of that. So keep in mind that all of these key innovations took millions of years to come about. Some have only evolved a single time, and we can, prove that with evolutionary relationships among animals. Some have evolved more than once, and so remember, if it's evolved more than once in a different lineage, we call it convergent evolution, right? Like shark bodies streamlined versus dolphin streamlined bodies.
What's a body cavity?
We'll get to that. I'll define it.
Yeah,
I mean, like, Body cavities are usually like areas inside of you that can hold things together. So you have multiple body cavities. You have a thoracic cavity, you have an abdomen cavity, cranial cavity. Okay, so we're going to talk about symmetry. We're going to talk about all of these, but first off is symmetry. So they either have radial symmetry or bilateral symmetry. Radial just means that you can cut it pretty much anywhere and end up with mirror halves. And it evolved first in marine organisms. We will discuss Cnidaria in this unit. So here's an example of radial symmetry. Now, you might want to make a note here. Sponges are the exception. Okay? Sponges have no symmetry. So that's why they're kind of in their own group. They don't really have true tissues. They don't have symmetry, so they call them parazoas. But I will, again, revisit that. So. All right, second type of symmetry is bilateral symmetry. So you cut it basically one way. You can only cut it one way. It has a left and a right half. I know you have a lot to write on this one. Bilateral symmetry is really important because now you have a top versus a bottom, and you have a front and a back. So you're going to see these terms when you get to dissection. Dorsal. I always think dorsal as back, like a dorsal fin on a fish. Bottom, ventral, that'd be the belly side. Anterior is usually towards the head region. Posterior, which doesn't look like I spelled it right, but bottom. And so with bilateral symmetry, we have this idea of cephalization. So cephala means head, and basically we're going to see a localized region of sensory. I mean, think about us, our head. We have pretty much almost all of our senses there. Sight, smell, taste, hear. Okay, it helps us guide us in our environment. So that's what cephalization means. So that's why it says greater ability to move, because you have all of those sensories in one area and it kind of guides you in your environment. Okay. Tissues, second thing. Second key innovation of animals. So tissues are groups of similar cells that carry out a specialized job. And going back to embryonic development, gametes fuse together in fertilization, you end up with a zygote. And those cells are basically stem cells. They are totipotent cells. That means that they can become any type of cell. They will go through cell differentiation to end up to be a muscle cell or a nerve cell or a skin cell. But once a cell differentiates, it is irreversible. So my skin cell doesn't have the ability to go back to a nerve cell. Here's another exception. Sponges. They can differentiate and dedifferentiate. So as a result, sponges do not have true tissues. So what do I mean by dedifferentiate? It means that, let's just say you have a cell in a sponge that lines the cavity where it feeds. So basically a cell that aids in digestion. All of a sudden, it can have the ability to lose that function and become a gamete. Okay? So that would be a great example of dedifferentiation. So our simplest animals are called parazoa, basically the sponges. They lack tissues and organs, but they do have cell specialization that are not found in protists. And so that's why we consider them animals, even though there are some exceptions. Other animals, which we will consider eumetazoa, distinct and well-defined tissues. Okay, body cavity. Third key innovation. So with a body cavity, now we have advanced organ systems. So we go from tissues, groups of tissues form organs. And during that embryonic development, your cells organize themselves into 3 germ layers, or maybe just 2, because there are some that are just diploblast. So there are 2 types of embryonic developments, you can end up as a diploblast or a triploblast. But if you are a triploblast, that means you have all 3 germ layers. And starting from the outside and working your way in, you have your ectoderm, your endoderm, and your mesoderm. And if an animal has all 3, triploblastic. Now, the ectoderm usually is the outer covering. So you can think of your skin, okay, epithelial layers. Your nervous system is also part of the ectoderm. The endoderm, think inside, so your digestive system, your intestines, and then your mesoderm, which is kind of in the middle, is going to be your muscles and your bones. So those layers will become muscles and bones, and then this will become digestive inner organs. Okay. Now, if an animal only has 2, that means they have an ectoderm and the endoderm, and they don't have the mesoderm. So cnidarians are diploblasts. Usually radial symmetry organisms are diploblasts. All bilateral organisms are triploblast. So with an efficient organ system, now our body cavities are isolated. They are surrounded by tissue. It gives them space. It supports the organs. It holds them in place. Certain areas carry out separate jobs, so we kind of have compartmentalization a little bit. So there are 3 kinds of body cavities. And so in our old college textbook, this is actually how they broke up the chapters. They did the coelomates, pseudocoelomates, and then the coelomates. This book does protostome, deuterostome, so the chapters are huge. So coelomates means they have no body cavity. So the flatworm here is a great example of an coelomate. And if you take a look at the mesoderm, which is pink, okay, and the endoderm, which is yellow, this space that's in between the mesoderm and the endoderm is basically filled with cells and organic materials. Pseudocoelomates, like the roundworm, you can see that their body cavity is not enclosed by the mesoderm. So their body cavity is a pseudocoel, so it's between the mesoderm and the endoderm. And then coelomates have the body cavity. So an earthworm, great example, which you will dissect in this quarter. Okay, now pseudocoelomates, they can move materials by churning fluids within the body cavity, but coelomates cannot. And so this problem, as you can see, is solved by the circulatory system. So the circulatory system is found in coelomates. They have networks of vessels. They carry fluids, waste products, gas exchange. And there are 2 types of open circulatory systems. 2 types of circulatory systems, open and closed. So open means that blood travels from vessels and then it will pool in a sinus area and then re-enter the vessels and go into another location. Closed means it flows entirely in blood vessels and doesn't pool in areas, and closed is faster and way more efficient than open. Okay, moving on to bilateralians. 2 types of developments. It starts off with, after the zygote, it goes through multiple rounds of mitosis, and it reaches an area of cleavage, and then it will end up as a blastula, which is a hollow ball of cells. Then the blastula will form a 2-layer thick ball called a blastropore. Basically, we have this indentation. And once that indentation occurs, you get a structure called an argentorion, which is primitive gut. And then the fate of this pore will determine whether or not you are a protostome or a deuterostome. So the protostome means that the mouth forms first, This includes flatworms, nematodes, mollusks, annelids, and arthropods. But if the pore becomes an anus, then it's a deuterostome, and you end up as either an echinoderm or a chordate, which is backbone. So proto means first mouth, deuterostome means second mouth. Now there are some other differences besides what the fate of the blastopore becomes. So I think I just have a diagram here showing cleavage, where 2 become 4 and 4 become 8. But take a look at when it reaches that 8 stage cell, that what happens to the orientation of the cells. So as it's becoming 4 to 8, notice that it's off-centered. Okay, and now you end up with like spiral cleavage. Or if it's right on top of each other, then you end up with radial cleavage. So we have cleavage patterns, which is what I'm trying to get to. So protostome versus deuterostome. You can also have differences in cleavage patterns. So in protostomes, it buds off at an angle and you end up with spiral cleavage. In deuterostomes, they are on top of each other, so then you end up with radial cleavage. So protostomes have spiral cleavage, deuterostomes have radial cleavage. Okay, some other patterns during development. So I'm still on the 4th key innovation here. Determinate versus indeterminate development. Protostomes, they have determinate development. So all of their cells are predetermined as to what they will become. If the cells are separated, development will still proceed. Deuterostomes have indeterminate. Basically, you have a first few cells that produce identical daughter cells, and then they will, I have a typo here, any one of these cells, if separated, can develop into a complete organism. This is indeterminate is actually, this is how identical twins can develop. So those cells get separated, but because they're still in that first couple rounds of division, they can turn into complete organisms. That's why they look exactly alike. Okay, formation of the coelom. So with protostome, basically the coelom is formed by spaces created by migration of the mesoderm cells to opposite positions, and in a deuterostome, they are formed by invaginations. So I don't know if you just wanted to summarize like gut formed by migration. Yeah, gut formed by migration. And then maybe just add over here, formed by invaginations, it folds in. I probably should just change that slide because I feel like that makes way more sense than what I just wrote. And then, do you want me to repeat that? Yes, okay. So protostomes, gut formed by migration of cells. So the cells move. And then deuterostomes it's formed by invaginations. All right, the 5th and key innovation of animals, segmentation. So with segmentation, you have repeated body units or redundant systems, and it aided in locomotion. So 2 big advantages. Each segment can go on to develop a more or less complete set of organ systems, which means that if any damage occurs to one segment, hey, that's okay. It may not be fatal because we have other segments that can carry out the same job. And then locomotion. Well, now you are made up of tiny little parts and you have more flexibility. So, different ways of locomotion could arise. Segmentation is mediated at the molecular level. I know we skipped over the chapter of Hox genes, but you see segmentation, Hox genes, in the majority of animals. All right, review questions. A blank is an example of a specialized organ system that assists with distribution of nutrients and removal of waste. Circulatory system. The innovation of segmentation allowed for redundant systems and what other important movement or improvement? Wow. C, locomotion. What is thought to have been the key innovation that fostered diversification in animals? B, specialized tissues. All right, this one's you gotta read through a lot. Select the correct array of defining features for protostomes. What? What'd you say?
Was it D?
It's not D.
It's B.
It's B, spiral cleavage. B as in boy. So spiral cleavage are protostomes. Determinate development. Blastiform forms the mouth. Coelom from the mesodermal split or migration of cells. Which of the following is not a key innovation in animal evolution?
E.
Yes, it is E. All choices are key innovations. Which innovation made possible the development of advanced organ systems? D. Body cavity. Yep, body cavity. Oh my gosh, it just keeps going.
E.
Yes, it is E. All choices are correct.
E again.
Select the correct
E.
statement concerning segmentation. Yes, E again.
Yes. There's more. D.
There's more.
D. D.
No.
Oh,
No.
sorry.
A fluid-filled cavity that develops completely within mesodermal tissue is a characteristic of A. Coelomate.
Yes.
Okay, animal phylogeny. So I'm going to talk about some of the major animal phyla, talk about patterns of convergent evolution, and where do humans place among the animal phyla. So there is some disagreement among animals in the phyla, how we arrange them. Traditionally, we just looked at anatomical parts or features as well as embryological development. But now that we have DNA sequencing, it has changed and resolved some relationships within the phyla. So this is the modern phylogeny. Okay, so let's just orientate ourselves. Here's Protista, the cyanoflagellates, right? And then we'll start off with sponges, which is Parazoa. And then this is where we have our true tissues, right? Our true animal kingdom here. So I'll talk about cnidarians and Ctenophoria. And then no body cavities. Then move into the protostomes. So Spirula. So we'll hit up all these. And then we have our Lophotrochozoa. That's a fun word to say. End it with the Ecdysozoans. And then chapter 35 is all about protostomes, the deuterostomes. So we'll hit the echinoderms and the chordates last. So, yeah. Question for you. Is the animal kingdom monophyletic?
Yes. Hold up.
I'd say so, yeah. Looking at this? Yeah. Yeah, I'd say. Okay, so our current understanding of phylogeny does differ from the traditional views. I wonder if I have a slide where I show you the traditional. Yes, I do, later on here. So it is a hypothesis, it's open to challenge, it can be revised with new data, but some consistent results is that the Porophora, or the sponges, constitutes a monophyletic group. Cnidarians branch off the tree before bilatera. Deuterostomes branch within bilatera, but it does have some inconsistent results. We have some protostome arguments, annelids versus orthopods, and how segmentation evolved convergently. Ctenophoria, both diploblastic and triploblastic, so that causes some confusion. Chitonathia is considered both a coelomate and a pseudocoelomate, and I don't know how that happens, but yeah. Okay. Based on morphology, if we were to create our tree, it's based on the coelom. Okay, so the traditional view is you start off with no body cavity, and then the pseudocoelomate, and then the coelomate. The coelom does appear to have just evolved just once. There is one clade within the coelomates that reverts back to acoelomates. Pseudocoelomates, there's multiple times where it arose, but all deuterostomes, chapter 35 organisms, they have a coelom, a body cavity. So. Okay, protostomes do include spiralians and the ecdysozoans. So those are the two clades that we'll start off with in chapter 34. Spiralians just means that they have spiral cleavage, and there are two organisms that you'll need to know, the Lophotrochozoas and the Plateozoas. So they move by muscular contractions, and Plateozoans move by ciliary action. That's kind of the big difference between those two. Ecdysozoans means that any animal that usually molts, and we only have seen this once, like evolved once in our phylogeny, and arthropods are the largest. You can see this cicada leaving its exoskeleton. It's molten. This is a lophophore, and over here is a zoom-up structure of the trochophore larva. I don't have a picture of the Plateozoa, sorry. Deuterostomes are your chordates and echinoderms. So they have fewer phyla and species than the protostomes, but they are way more uniform in their appearance. So.
Wait. Brain,
Why didn't you tell us?
I was not ready. Alright, thank you. Since he's catching us slacking, man, tighten it up there.
All right, so here's what the old phylogeny tree looked like based on morphological and developmental comparisons. Sponges, and then we hit the true animals, and then you would start off with cnidarians, and then tenetophores, and then it branches into your bilateralarians. And then you hit up your deuterostomes and protostomes. This is what the new tree kind of looks like based on molecular. And the words change, but you still have your sponges, and then your cnidarians and tenetophores, then your bilateral, and then they break it up into deuterostomes. So there's your two right there, echinoderms and chordata, or chordates. And then your protostomes, Lophotrochozoas and ecdysians. So this table right here I feel like will be of great help for your guided notes this week. It's pretty much got all the major phylums, examples, some key characteristics, and how many species are located in each. And will we go through all of these? Pretty darn close, not all of them though. So, okay, which of these is not a protostome?
C.
B.
D, echinoderms. Do you just have like a number, like a letter in your head? You're like, I'm going to say C for the next one. What has recent molecular data revealed about the phylum Paraphora?
D. No.
None.
No. D.
It's A, it's monophyletic. I'm pretty sure I have that in the notes. Recent molecular data has led to a revision of the traditional classification of animal phyla. The annelids and arthropods are now moved into different clades, and the arthropods are now assigned to... Yes, it's B!
Cole says true.
It is accepted that the animal kingdom is monophyletic.
True.
True.
Good work, Cole.
What do deuterostomes not include? Not include.
No, deuterostomes have chordates.
It is actually B, ecdysozoans. Those are the ones that molt. So yes, chordates are part of deuterostomes. Echinoderms are. I didn't talk about examples of echinoderms, but sea stars and sea urchins are echinoderms. Okay, which clades do Spirilia comprise? E, yes, the Lophotrochozoa and the Plateozoa. Okay, moving on to the Parazoa. Sponges. Okay, very loose body organization here. Few that are radial symmetrical, but most of them are asymmetrical. Larval sponges are free swimming, so they can swim in their larval state, and then when they reach their adult form, they are anchored, and so now they are considered sessile. Sponges can defend themselves by producing chemicals to repel predators or organisms that might try to overgrow them. All right, we do see several different cell types in the sponge body. Okay, so sponges, they have no head, they don't have appendages, a mouth, or an anus, but their cell structure is somewhat organized. They have cell types with different jobs like contracting the oscula, which is the pore, synchronized reproduction, and they do have an organized meshwork of spicules and spongin, which I'll show you on the next slide here. So, you know, sponges, basically, they're just a mass of cells embedded into some type of gelatinous matrix. Remember, our cells can de-differentiate from a specialized state to an unspecialized one. And they've done experiments where they've taken a sponge and they've shoved it through a sieve or a coarse cloth, basically like a mesh, right? So all the cells separated from each other, and those cells will actively seek one another and reassemble the entire sponge. Okay, their walls are made up of 3 layers: cyanocytes. And you're probably thinking, hey, cyanoflagella, right? So here's what a cyanocyte looks like, and it is very similar to cyanoflagellates, where they have a collar, they have a flagella, and that flagella beats and moves water so food particles can get caught in the collar. And then it does phagocytosis of the food particles. So the cyanocytes make up the osculum, and that is where it is water is expelled. So you can kind of see these cyanocytes lining the inside, and they wiggle their flagella, and water will come into the sponge and then exit out the top. Okay, we also have some pores. So an outer epithelial layer of flattened cells, as you can see here, they're called, looks like porocytes. And then the mesohyl, which is kind of this yellowish matrix going on here, where we see spicules, which are yellow, and spongin, which I would assume is the kind of light orange area. So, yeah. They can reproduce asexually and sexually. Asexually just means they break off into fragments and they can become entire organisms. Sexually, they have sponge sperm created by the transformation of cyanocytes, and they release them into the water. Sperm is captured by a cyanocyte, carried to an egg cell somewhere in the mesohyl, and then development occurs on the mother, and then other species will release that fertilized egg into the water. And then remember, once it reaches adult form, it is sessile. Okay, how do sponges differ from all other animals?
B.
It is B, they lack specialized tissues.
I thought they can go from.
It said. Hold on. They are. Wait a minute.
In their adult form, but not the larva state.
It's they have,
They can de-differentiate. That doesn't say
it's
de-differentiate.
cell types with specialized functions.
I thought it said they could differentiate.
Yeah, so that is why it's B. They lack specialized tissues because their tissues can become anything.
Oh, wow.
I'm glad we're clearing this up because this is, yeah.
Yeah, man, I get you now.
All right.
Picking up what you're putting down.
Alright.
Okay. We good?
Yeah.
Okay. Which of the following is characteristic of sponges?
Well, yeah.
We don't have
They can
symmetry.
sexually
B,
produce.
They don't have symmetry, tissues, or organs, or mobility.
Oh, you know what? I think there's two answers here.
Well, B's
B
one.
and E. You need.
There's no donor.
Well-
They don't move. What
Is
is
it C and D?
Can't they move?
It would be B and C. Well,
I think I actually might have a typo here. I think A is supposed to say asymmetry, and I just forgot the A because it's A, option A.
Symmetry.
Yeah, I'm gonna fix that. I'm pretty sure
Asymmetry.
asymmetry. Sorry.
Hey.
don't sponges
Hey.
have?
Sexually reproducible.
Depending on how you look at it. So like here's a sponge, right? You could say, oh, there's radial symmetry there, but like, are you looking at the entire organism, or are you just looking at one little tube thing?
Sorry.
Okay, select the statement that is not true concerning cyanocytes.
Not true.
C.
Yes, it is C. They consume particles many times their size by stretching their cells. So basically, they can only eat things that are smaller than them. Okay. Spicules and spongin are found blank of a sponge.
So B.
B within the mesohele. All right, so that's the parazoa. That's sponges. Moving on to the eumetazoa, animals with true tissues. So we will kick off this section with the cnidarians. So the first key transition, remember, was distinct tissues, how we have different cell layers, our ectoderm, our endoderm. Now, cnidarians, they are dipoblastic. They are, any animal that basically has radial symmetry is dipoblastic. So they have distinct tissues but no organs. So there are two radially symmetrical groups that exist today, the cnidarians, which include hydroids, jellyfish, sea anemones, corals, and then your Ctenidophoria, which include the comb jellies.
What are comb jellies?
Comb jelly.
Oh yeah.
Yeah.
But what are they?
They're just comb jelly.
They are very translucent. They have
Friday.
these two distinct tentacles. I mean, you would think of them as, you would look at them and think that they're jellyfish, but they're slightly different because their two big tentacles are like ribbed. I don't know, it's kind of, I don't know. Not what I said, but okay.
They live like funny.
Okay, so the basic body plan of cnidarian, gelatinous in composition. They are carnivores, capture and eat in their prey with tentacles that ring the mouth. Two basic body plans shown in the picture here, the polyp and the medusa. So the polyp is usually attached to a substrate. They have an open end that acts as both mouth and anus, and it faces upwards. So it's got these tentacles, and they capture their prey and bring it into the mouth. They do their digestion, and then they excrete the waste back out through the mouth. Medusa is kind of the free-living, umbrella-shaped thing with tentacles. Kind of floats here. They don't have any really reproductive system, circulatory system, digestive system. We'll talk about how they digest their food here. Excretory systems, but they can reproduce even though there's no reproductive system. They can do gas exchange even though they don't have a circulatory system. They can capture and eat their prey and distribute those molecules even though they don't have a true, circulatory system. So yeah, let's talk about that here. So here's the polyp versus the medusa again, showing the cross section of it, if that's how you want to think of it. I'm going to be talking about the mesoglea here on the next slide. But here's a sea anemone, which is considered a polyp, and then here's a jelly, which is considered a medusa state. Okay, so in their gastrovascular space, and I'll go back after we get the notes here, this is where digestion, gas exchange, waste discharge, how the gametes form. It also serves as its hydrostatic skeleton. And basically this area gives the animal its shape. It's made up of 2 layers, the epidermis and the gastrodermis. So the epidermis means outside the environment, gastrodermis means inside the gastrovascular cavity, and then between the epidermis and the gastrodermis is the mesoglea. So let's point out the gastrovascular cavity. Okay, so here it is right here in the polyp. And then you can see that it is lined with the gastrodermis and then the epidermis. And then in between these two is the mesoglea. Over here with the medusa, here's our gastrovascular cavity, and it is lined by the gastrodermis and then the ectodermis, which is just another word for dermis. And then in between here, the mesoglea. Life cycle. Some only occur as polyps, some are only medusas or medusae, and some can actually alternate between the two. But just keep in mind that everything here is diploid. Polyps, they reproduce sexually or asexually. If it goes through asexual, it can be a polyp or a medusae. They do have a larval stage called the planulae. Medusas can only do sexual reproduction. Okay, cnidarian digestion. So everything kind of takes place in that gastrovascular cavity. What it does is it has digestive enzymes that are released. They break down the food. Then the cells that are lining it will use phagocytosis to bring those food particles in. And because it can capture and eat its prey and kind of digest it, break it down, they do have the ability to eat animals larger than those cells. So remember with sponges and cyanocytes, they can only have food particles that are small enough for the cyanocyte to engulf it. Now we have an animal here that can consume a prey larger than a sponge or than a sponge can. Okay, another unique thing about cnidarians is they have specialized cells called cnidocytes. So here, this entire thing here is a cnidocyte, and within the cnidocyte you have something called a nematocyst, and it's this coiled up harpoon-like thing. And a trigger sets it off, shoots out, injects into the prey, usually toxins that will subdue the prey, paralyze it, and then it will bring it into the mouth, break it down, and then, yeah. So this is why jellyfish have that sting. It's because of the nematocysts. There are 5 classes of cnidarians. Class Anthozoa, sea anemones, and corals. So these are plant-like. They are the largest of the cnidarians. Their tentacles are hollow. Coral reefs are part of class Anthozoa. Refuge for crustacean fishes. Yeah, refugees, yes. Right now, with oceans becoming warmer, we have coral bleaching that's occurring. So as carbon dioxide levels rise, dissolves in the water, makes the water acidic. The acidic water breaks down the calcium carbonate, so there's less of it available. So coral reefs are disappearing. Second and third class of cnidarians, Cubozoa, or the box jellies. So you can see that they have a box-like shape and then groups of tentacles hanging from each corner of the box. Hydrozoas, or the hydroids, they have both a polyp and medusa stage in their life cycle. The polyp stage is colonial, as you can see down here. Some are bioluminescence. This is the Portuguese man of war. It's a floating colony of highly integrated polyploid and medusaloid individuals. I don't even know if those are words, but that's how I would describe it. So
Which one's that?
this one right here. Okay, the fourth and fifth class. Class Scyphozoa. I'm butchering that name probably. The jellyfish, essentially colorless. And then the last one, this is kind of a new class in this book. Our old book did not describe the star jellies, but class Staurozoa, the star jellies. So it's separated from our Scyphozoas. They look like Medusa, but they are attached to substrates. So these two right here are jellyfish, and this is our star jelly. So we just went through the Cnidarians, and we will pick it up tomorrow with the Ctenophoria.